Hello and welcome. Starting from today, I'm going to devote a couple of sessions for a very important disease of humans we know as cancer. According to one study of WHO, cancer is the leading cause of death worldwide, accounting nearly 1 in 6 deaths. So that shows the seriousness of the disease, which is acknowledged globally. And therefore, a lot of work has gone in last 4 or 5 decades to understand all aspects of this disease, which has resulted into giving us some understanding about its cellular and molecular basis. So in this session, very specifically, I would be looking at what goes wrong in a normal, healthy human cell that it becomes cancerous. And also, we will see what is responsible for that change from healthy, normal to a cancerous cell. And towards the end, I will talk about some factors which contribute towards the development of cancers. Okay, to begin the discussion, let us start by giving a working definition to this disease we know as cancer. What is cancer? So basically, in cancer, a normal cell undergoes a change in behavior, and this behavior is with respect to its division. Now, what kind of change it undergoes? That is, it starts dividing without any controls and regulation, and it becomes immortal. And finally, this dividing cell starts spreading to other parts of the body. Okay, the process we call metastasis. So this is a broader definition to cancer. What happens in cancer? A normal, healthy cell changes its behavior, that it starts dividing, becomes immortal, and then the cells starts moving to other parts of the body. Okay, now to understand this phenomenon of a cancerous cell, we must know how a normal cell behaves inside the body of humans or human beings. What kind of controls a normal cell has. If we understand this part, then it becomes easier to understand that what may be happening when a normal cell becomes a cancerous cell or starts behaving differently with respect to division. Okay, so let us see what happens in the mature human body cells. You may recall that cell division is a process which differentiates a living cell from dead cell. Dead cell never divides. So it is one of the properties of living cell that they divide. They can undergo division. Okay. But what happens in a mature human body when the cell undergoes differentiation and organ formation? These cells, they stop dividing. And even if they are recalled back for division or for dividing, this process is highly controlled and regulated. For example, what I'm trying to say is that cell division is a process which is required for normal growth and developmental process, for their maintenance, you know, of an adult body, you know. But then this division of an adult mature cell is highly coordinated in terms of timing when a cell has to divide, and also how many times a cell has to divide, you know. And also what is seen that not all cells have the same cell cycle. I will come to this part a little later, but then the point to be understood here is that in a mature human body, the cells normally don't divide, but even if they have to divide, it is a very, very coordinated activity in terms of when they have to divide and how many times they have to divide, you know. So this, keep this in mind, you know. And also, see, for example, what is the frequency of cell division? You know, for example, what we know today, that a embryo cell, it divides every 20 minutes, because at that stage when the uh, the 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 child development is taking place in the womb, after fertilization, the cell first divides, multiplies millions and billions of times, and makes a mass of cell. And then the cytodifferentiation takes place and the developmental process takes over, you know. But then in a mature human being, if you see here, a skin cell, for example, it divides once in 12 to 24 hours, you know. So that is the frequency of a skin cell. And why they need to? Because of the maintenance purposes. On the other hand, if you see liver cells, they divide once or twice in a year, you know. And interestingly, nerve cells and muscle cells, they never divide. They are permanently in what we call G0 phase, you know. So again, with this kind of information, what one is seeing, that the cell division is highly controlled and restricted process, you know, in a mature human body. You know, it takes place only when it is required, when human body wants any particular cell, and that too few cell types, not others. Others never divide, you know. So for example, the cell cycle I was referring to, that not all cells in the human body, those who are dividing, follow the same cell cycle. So basically, this is the cell cycle one was referring to. So you have a M phase, you know, where the one cell becomes two, but then before that happens, the cell has to undergo a period of rest, and this period of rest is divided into three phases: the G1, S, and G2 phase, you know. And these are very important phases, and we know, and I will talk about these phases in a while, what goes on here actually. So this is the cell cycle one is referring to, that all the dividing cell inside the body, they are not on the same plane, you know, that not all of them are in G1, S, or G2 phase. They may be at different phases of this cell cycle, you know. Okay, so now if we say that the mature human cell inside the body is highly controlled and regulated, and this is the cell cycle, it follows that after the cell becomes two, 1 to 2, it undergoes a period of rest, and then again it divides. Fine. So what are the checkpoints? So in this cycle, we now know, I'm just going to summarize for the sake of understanding what happens on cancer cell, you know. I mean, this is the general agreement that there are three checkpoints during this cycle where the cell commits that yes, it has to divide, or it doesn't have to divide, you know. So first checkpoint, if you see, is the one, the G1S. After the cell enters into the G1 phase, so at this point, it commits itself that whether, yes, it has to divide, and it enters into this phase, the second phase, that is synthetic phase, So this is the first checkpoint, where a cell decides, and there are certain parameters which cell has to understand before it commits that whether that has taken place or not, you know. So, and this is also called the restriction point. Okay, the first restriction point means the question the cell answers is whether the DNA synthesis can begin or not. So before that, cell has to prepare itself in terms of enlarging in size, accumulating a lot of resolves, etc. And then the next is this. Next checkpoint is the G2M checkpoint, where where the cell tries to find out whether the DNA synthesis has been completed and completed correctly. So that check happens at this point, and then it commits itself to enter into the mitosis process or cell division process. Okay. Now, third point of check is the spindle checkpoint. So this is the third one, where the cell checks whether all the chromosomes are attached to the spindle and the sister chromatids can separate correctly or not. So this is the third point. But major controls are these two, G1S transition point and G2M transition point. So these are the three major checkpoints. And of course, the spindle checkpoint, So the point I'm trying to make here is that if the cell finds out at any point out of these three that it is not ready, for example, if it is not ready for DNA synthesis, it will stop here. Or if the DNA has not been synthesized correctly and completely, it can stop here. Okay. Now, these stopping and even starting a cell to divide, there are well characterized chemical signals which are released by other cells and perceived by the dividing cells, and in response to those signals, the cell decides whether it needs to divide or not, or it checks through whether the preparations for entering into the next phase is completed or not at this point and at this point. Fine. I'm not going into the details of those chemical nature of those controls because that is not the point of discussion here. Okay. Now I was talking about external signals, and they are mainly growth factors. Now, growth factors are the one which coordinate or has coordination between the cells, and they stimulate the body cells to divide. Now, there are two things which again you have to keep in mind, and we will look at it later when we talk about cancer cells. That a normal cell shows, this is one of the properties of normal cell when it is dividing, that it is density dependent inhibition, or the cells show density dependent inhibition. What it means? That when the dividing cell becomes crowded, they stop dividing. Why? Because the growth factor signals, they do not reach to all the cells or the dividing cells, so they stop dividing. And second property they show is that they are encourage dependent. The dividing cells are encouraged. They need a substratum to attach themselves, in normal cells. So normal cells show these two properties, that is density dependent inhibition and the encourage dependence. And this is because of the touch sensors. All cells have the touch sensors. So they give them signals that whether they are touching some substratum or not. Okay. So So to conclude this part, what I have tried to make you understand that how a normal human cell behaves inside the body with respect to the dividing. They know when to divide, how much to be divided, and this is helped by various growth factors and signals uh, which are released or gets activated during the cell division cycle to stop to initiate the division or not to initiate the division. Okay, so it is very important phenomenon for a normal cell to behave, to know when to divide and when not to divide, and how to keep these cells which are not dividing in the non-dividing state. You know, okay, now with this kind of background, let us come back to look at how a cancer cell differs from a normal cell. Okay, I gave a broad definition to begin the discussion earlier, but now let us look at uh, with this background which I gave, that what happens. So on a normal cell or normal human cell typically has this kind of cycle. That is, they grow, divide, and die. Means a cell which is which gets damaged, you know, it dies by itself. So there is a mechanism what we call apoptosis or cell death or suicide. So if a cell, it will grow, it will divide, and remains in, if it is remains healthy, it is fine. If it is not, then it kills by itself. So this is the normal cycle which is followed in a in a healthy human being. But then what happens in cancer? You know, it does not follow this cycle. Okay, it does not follow this cycle in the sense that it starts growing in absence of signals telling them to grow. I told you a normal cell will grow and divide only in response to certain chemical signal, what we call growth factors. But a cancer cell, what happens? It starts dividing or growing in absence of any signal. So that is one difference which happens in the cancer cell. And secondly, which is all the more important, that these cancer cells, they start ignoring signals that normally tells a cell to stop dividing. You know, I told you that there are signals which tell a cell to stop dividing when it is not needed. Okay, but the cancer cell starts ignoring those signals, and also it ignored the signals uh, which are responsible for its death, what we call apoptosis. Means the cancer cells, they uh, they ignore the stop signals as well as signals which tells them to die in case the cell uh, is not healthier enough to continue, which happens in a normal cell. So in cancer cell, this thing is happening. And lastly, when these cells, multiply, they move to other parts of the body, the phenomenon which we call metastasis. Okay, so basically, these are the changes, behavioral changes, which a healthy cell undergoes when it becomes cancerous. Okay, now we will see how and what is responsible for these changes. Okay, and what is the consequence? First, let us see what are the consequences of this kind of behavior. So, what happens? You know, when these cells undergo uncontrolled division, they form a mass of cell, okay, what we call tumor. So these tumors are basically of two types. One is benign tumor. you know Benign tumors, basically they remain there, at their site of origin, basically. They don't move, and therefore they are not a serious threat to the human health, basically, and they can be removed surgically, and that is taken care of. But the other kind of tumor, which is we call malignant tumor, you know, here the cells leave the original site. Now that is the problematic. That is, they lose attachment to the nearby cells, and these cells are then carried by the blood and the lymph system to other tissues, and they start, uh, you know, tumor formations in other parts of the body, and this process we call metastasis, and they wherever they go and form tumors, they impair the functions of that organ of the body. Okay, so basically, the consequence of a normal cell becoming cancer cell is this: it forms tumors which are of two types, benign tumor and malignant tumor. Benign tumor are less serious because they normally remain at the original place. They don't move out and can be removed surgically. Whereas these malignant tumors, they are a serious problem actually because of their nature or this property which they have, that of a metastasis. Fine. Now, okay. Now, growth factors, how growth factors are responsible for cancer. Okay. Now we are going to address the question actually, that what is responsible for this change in behavior, which I referred from normal to cancerous cell. Okay. So I told you a cell starts dividing when it should not divide. Okay. And there are uh, growth factors which are responsible for telling a cell to divide. Okay. Now, so there is one set of genes we call proto-oncogenes. Now, proto-oncogenes basically are normally genes which are responsible for activation of cell division in a normal cell. So basically, they are growth factor genes. Okay, growth factor genes, and these genes express themselves only in dividing cells. Okay, not in non-dividing cells. When the cell stops dividing, these genes don't express or stop expressing. But then what happens in cancer? Uh, these these proto-oncogenes, they express themselves. How? When the genes gets mutated. If some mutation occurs in these genes in such a way that it starts expressing, or what in scientific term we say that if they are switched on. So switching on of these proto-oncogenes, which normally are responsible for cell division, they cause cancer. So in a normal healthy cell, these genes don't express, but in cancerous gene, they start expressing. So one reason of cancer development is that when certain genes which should not express in a cell starts expressing. Okay, and how they start expressing? By way of mutations. There are many ways by which a gene can undergo mutation, and I will take up this issue in my subsequent videos. But for now, you can understand what's going on here, that a normal, we call it as proto-oncogene, means it can become cancerous, or it can become oncogene, oncogenic. Okay, so it is a prototype of a copy of a cancer gene actually. But in a normal healthy cell, it has a function to perform, to make a cell to divide, but then it is all under strict control. It knows when to express. So whenever there are growth factors signals received by a cell, these genes start expressing, but then what happens when these genes start, they get mutated, in such a way that they start expressing constitutively without any control. That is again. So a normal gene is under heavy control, but a mutated gene loses that control and it starts constitutively expressing itself. Okay, so this is one set of genes which is responsible for causing cancers. Okay, there are more than half a dozen of such genes have been identified and characterized from different kinds of cancers. For example, RAS is one which is responsible for activating this component of the cell cycle, which is responsible for the checkpoint clearances at G1S and G2M. Okay, so those mechanisms I'm not talking, but just to give a reference here, this is one such gene, which is a proto-oncogene, which is responsible for taking the cell division in as and when it is required, but then, because of mutation, it starts expressing and activating this component, and the cell starts dividing in an uncontrolled manner. So this is one part of it. Okay. Another set of genes we call tumor suppressor genes. Okay, tumor suppressor genes. So, as I mentioned, uh, that human cells, they know or they remain in a non-dividing phase, and they only divide when they are made to divide, when they are asked to divide. So these genes, what we call tumor suppressor genes, are the one which are responsible for keeping a healthy cell in a non-dividing phase. Okay, non-dividing phase. So therefore, tumor suppressor genes, their expression in a healthy cell is required. Their expression in the normal cell is required so that the cells remain in a non-dividing phase. Okay. But then what happens if these genes, tumor suppressor genes, their expression is stopped? Or in other words, we say that if the tumor suppressor genes expression is switched off, okay, then it causes cancer. Okay, it causes cancer. So you can understand what's happening when something is responsible for not allowing cell to divide and that stop functioning. Then what will happen? That control will go and the cell will start dividing. So that check is gone, which keeps the cell in a non-dividing phase. Okay, so we call them as tumor suppressor gene because they suppress tumor formation. If these genes are not there, then the control goes and the cell will continuously be dividing. So their expression in all the mature cells of human is needed. And how this is switched off, how the expression is switched off, again by way of mutation. Means these genes undergo mutation in such a way that they stop expressing themselves. And when they stop expressing, the control also goes. Okay. And one example, which is very well studied in almost all cancers, that is P53. P53 is the gene which is one of the tumor suppressor genes, and a mutation in this gene is responsible for a variety of cancers in humans. Okay, and I will spend some time how this gene functions basically. So to summarize, the genes responsible for cancers, I have talked about two types of genes. One is proto-oncogenes, and another is tumor suppressor genes. If you recall, I mentioned proto-oncogenes are the one who do not express in a non-dividing cell. They do not express in a non-dividing cell. They express only when a cell needs to divide. Whereas the tumor suppressor genes, their expression continuously is required to keep the cell under non-dividing state. Okay? Okay, fine. If you have understood, let us move forward. So what happens, cancer and cell growth? So as I mentioned, these are the two stop points, the G1 checkpoint and the G2 checkpoint. Okay, and this is the M checkpoint. That is the third the control point. So basically, in cancer, what is happening that it is it loses the control over its cell division. Okay, so this loss of control over cell division can be at this point, or this point, and therefore it undergoes uncontrolled, unrestrained cell growth. Okay, and basically, what control is lost in this? These checkpoint stops. So, in the cell division, when a cell starts dividing uncontrollably, these stop controls are gone. The cell doesn't know when to stop and where to stop. So it starts continuous division, So this checkpoint stops, control is lost. And as I mentioned, this P53 plays a key role, basically. And it is involved here in the first control point, that is G1S transition point, or restriction point I mentioned. This gene product is involved here, Now, this gene product basically stops or halts the cell division at this point if it finds, that dividing cell which wants to divide, its DNA has been damaged. If a cell's DNA is damaged and if it is trying to divide, this P53 stops that cell to divide, and it has a couple of options, to perform. What are those options? That it can stimulate a DNA repair system so that it can fix the DNA which is damaged, or it can force the cell to go into the resting phase, or not allowing it to divide. It can make the cell to be in a G0 phase or the resting phase permanently. Yeah. Okay, and also, if it is not possible to stop it in G1 at this point or at G0 point, it can cause the damaged cell to die, or we say it induces apoptosis in the cell, or it asks cell to commit suicide. Okay, so this is the normal P53 does. When it finds the dividing cell DNA is damaged, it first tries to repair it. If it is not able to repair, then it causes cell to die. Okay, and what is seen, interestingly, that in all cancers, this mechanism or P53 activity has to be shut down. So this gene expression is stopped, and therefore, whether the dividing cell's DNA is damaged or not, it just starts continuously dividing, with whatever fault it has at the DNA level, So this is the main culprit for almost all cancers, which have been studied and seen that that P53 activity shuts down, So as I told you, in a normal cell, this gene needs to express itself, okay? But in cancer cell, this gene is not expressing. Okay, why it is not expressing? Because of some mutation. Some mutation has taken place and it has stopped expressing itself, or expressing it even if it is made, it is not functioning properly. Mutation doesn't mean that it will not express at all. It may make some protein, but it may not be functioning properly. So in both the cases, if it is not made completely, or some faulty protein is made, it is not able to perform its function what it should. Okay, let us look at more clearly what's happening, here. For example, in a normal scenario, what I was trying to tell you, with the help of this cartoon, let us understand what is happening. Suppose in a normal cell, if a DNA of a cell is damaged, okay, and the P53 protein it detects that some damage is there. So what it does, it tries to repair this DNA, and and then it allows cells to divide. Means after repair, the DNA has been repaired and now it can divide. But then if this damage is not repaired, then what happens? It stops the cell division. Okay, and then these damaged cells are induced to for if the DNA damage is beyond the repair, then these cells are induced to die or commit suicide, or uh, the mechanism we call apoptosis is induced into these cells and they die. So this is the normal behavior of this P53 gene, that if it first it will try to repair, and if it is not, then it will trigger the destruction or the damage, destruction of the damaged cell. But then what happens when this P53 is abnormal or is not there in the cell by way of mutation? Then what happens? That it fails to recognize the damaged part, DNA, number one. Okay, it's not able to recognize this protein here, I mean the damage in the DNA. Okay, and then what happens? The cell continue to divide, with this damaged DNA, So repair is not taking place, and when these damaged DNA the damage is accumulated, the damages in the DNA accumulates to a level that it becomes cancer. It becomes a cancerous cell. Okay, so all human cells, they have a mechanism inbuilt to take care of these kind of damages taking place in DNA, which is very normal process or feature of a living system. But then, the mechanisms for repair is there, but problem comes when these mechanisms don't perform their function because of the mutations in their respective genes, like one P53. So P53 has been regarded as master regulator gene basically for cell division, because it stops a cell to divide or remain, keeps all the human cells, non-dividing cell, in the state of non-division, Okay, so now to conclude, I took one example of P53, tried to explain to you how a tumor suppressor gene controls the normal cell growth, and what happens when it undergoes a mutation that it stops functioning normally, and a normal cell becomes a cancerous cell. As I mentioned, a lot of work has gone in, and we have now a large number of proto-oncogenes identified, which are involved, for example, genes involved in unlimited growth and immortality and blood vessel growth. These genes, these are all proto-oncogenes, genes for unlimited growth, immortality, and blood vessel growth. They are proto-oncogenes and which are switched on in a cancerous cell. Normally, they don't express, but in a cancer cell, they start expressing themselves and and giving this phenotype to the cell. You know, and in a normal cells, generally the genes which are responsible for these three features are not expressing themselves. You know, but you have other features, you know, which are regulated by tumor suppressor genes like ignoring checkpoints. I mentioned P53 is one example, which normally is expressing itself in all the cells, but in a cancer cell, its expression is stopped, or it is made abnormally, or the protein is abnormal and it lacks its functionality. Okay. Similarly, there are genes for apoptosis. Now, these genes are normally expressing. It is also the genes responsible for apoptosis are also tumor suppressor genes because they allow or they make a damaged cell to die or to commit suicide. Okay, so uh, so this is also a category of tumor suppressor genes which are expressed in a normal cell, but um, but are turned off in a cancerous cell. Okay, so and then yeah, they are also responsible for overcoming the anchor and the density dependence of the normal cell. I told you these two properties which normal cell possesses, that they require anchorage or substratum to grow, and they are sensitive to density or the crowding of the cells, dividing cells. You know, so these combination of the genes are also responsible to overcome this, so that a cancer cell, it can make a tumor, it becomes crowded, still it keeps on dividing, you know, and also they don't require any substratum to grow, you know. So to conclude, you know, what we have tried to understand, that a normal healthy human cell, uh, when it undergoes a behavioral change with respect to division, and how it undergoes this change, we have discussed that part, and I have tried to explain, taking few examples uh, uh, to make you understand that what happens when, for example, a tumor suppressor gene is not made or is not expressing by way of mutation, then cancer cell, and tumor suppressor genes are switched off, you know, to make it a cancer cell, you know. So fine. Uh, so these are some of the factors which may be responsible for making or converting a normal healthy cell to a cancerous cell. You know, these are the some of the known mutagens which can cause mutations in the genes: UV radiations, chemical exposures, radiation exposure, heat, cigarette smoking, pollution, age, and genetics. All of them have been found to play a role uh, in causing mutations in the genes. You know, so all are potential factors which can cause cancer, basically. You know, so I will be in my future videos. I will be talking about in detail some of the cancer types, the current status, where we stand in terms of treatments, remedies, you know, and I will be definitely looking at the age as one factor which is responsible for cancer developments, and genetics, how genetics contribute in a major way for cancer development in humans. Okay, so with this, I stop today's discussion, and I hope you enjoyed listening to this video as much as I enjoyed making it for you. So thank you so much. And next video would be on aging and cancer, how aging is responsible for causing cancers in humans. Thank you very much.